Today we're going to talk about how clean is too clean, and this is in the context of our bodies and health and disease. And we're going to talk a lot about microbes today and how diverse they are. We're going to focus in on that because microorganisms are fundamentally different than human beings and animals and plants and things that we see around us every day. We're going to talk about a clinical problem called inflammatory bowel disease. Chances are that in a course this big, in a class this big, that there are some folks in the some students taking this class that actually have IBD, and we're going to talk a little bit about that in the context of the environment and microbes and how we interact with those. And then finally, we'll talk a little bit about biotechnology that we use to study gut diseases like IBD. These include germ-free and gnotobiotic animals that we have right here at Montana State, and some interesting tissue models that we use in the lab called intestinal organoids. Okay, so microbiology, or the study of microorganisms, really has changed in modern times in the past, I'd say 20 to 30 years. Typically, microbiologists would look at the world or look at microbes through a microscope, shown there on the left, or by culturing microorganisms on agar plates, or just solidified plates like Jell-O, seen on the right. And you can see that these bacteria that grow up over here on this plate, each of them started theoretically from a single cell, and they made these beautiful looking colonies. So either we would look at them directly with a microscope, or we would try to grow them and look at them on a plate like this. But those two things have recently changed, again, in the past 20, 30 years. And now we look at microorganisms primarily through their DNA sequence and what genes are in their genomes. And this graph over here, it's a little bit dated, but it just goes to show how many microorganisms we know to exist based on their DNA, and that's called environmental clones, or whether we can culture them in the laboratory, and that's the cultured bacteria. So you can see, hopefully, as the years here progress from 1996 to 2005, many more microorganisms were discovered just simply by looking at their DNA versus culturing them in the laboratory. And that's important because we don't really know everything about microorganisms, what they do, for example, what enzymes they produce. All we know about them for the vast majority of microorganisms that we know exist is their DNA sequence. But that brings up an issue by studying them, and that being how diverse, what do they do? Microorganisms are everywhere. They're in things that we touch, they're in water, they're in soil, they're even in the air. And they're really not like us. And that means that we can't see them, we can't detect them with our five senses, right? We can't touch them, smell them, hear them, whatever. So we really have to go on the scientific method to study these things. And we can't just look out, say, as the picture here illustrates, look out on the Serengeti or something and watch these organisms interact and see what they do. We have to use the scientific method, and we need to basically study things that we can't detect with our own senses. And DNA sequencing helps that. And so here's an example of how DNA sequencing was used back in the late 70s, early 80s to generate what we call the tree of life. Okay, so this tree represents all living organisms on Earth, and this tree was made simply by comparing the genetic material in living cells. So over here on the left, you see the bacteria. In the middle, you see the archaea, and on the right, you see the eukarya, and that's where humans and plants and animals are found in this tree, and you can see those tips labeled. And I just want to point out that these branches in this tree, how long these branches are, corresponds to how genetically similar these organisms are, these cells that were used to conduct this analysis. So you can see that animals, plants, and fungi all kind of group together very closely in this tree, and that they're very different than, say, bacteria and archaea. In fact, most of this tree is actually microorganisms, and that's how diverse they are. And you can see here that animals, plants, and fungi are generally closely related compared to all these other things. Now, another way to talk about diversity is just comparing the genomes of similar microorganisms. We've all probably heard of the bacteria called E. coli. So E. coli is in 90% of people. We carry it around normally in our gut microbiome. It's a healthy microbe to have in us. We call it a mutualist. But anyway, when E. coli first began being studied with genome sequencing, there was a study that came out comparing the genomes of 3 different strains. And you can think of these as like 3 different individuals of this species of E. coli. And these genomes were compared. They found all the genes, and then they said, okay, how many genes are shared among these 3 strains? And that's what's shown here in the middle box. These 3 individuals of E. coli only shared 39.2% of all of the genes in their genomes. And that's remarkable, right? Because any individual in the human population, or in our species, Homo sapiens, we would carry all the same proteins, but we would have a few genetic mutations in those proteins that would differentiate us. Okay, so pretty much 100%. If you did this same analysis with the humans, that middle box would read pretty much 100%. And in bacteria like E. coli, it's down at 39%. So that just goes to show how diverse these individuals are, and what different things that they might be able to do. Okay, so what are we really? This slide is just meant to illustrate that really the cells that make up our body are in a minority. Okay, so the cells that make up our body are outnumbered pretty much 10 to 1, between 10 to 1 to 100 to 1 by microorganisms. Okay, and this just goes to show throughout our gastrointestinal tract, we have microbes that live in our stomach, in the small intestine, and in the colon, or the large intestine. And you can see these numbers get really, really big. CFU is just cells or colony forming units per mil of content in the gut. And microorganisms aren't just in the gut. We have microorganisms in our lungs and on our skin. Pretty much anything in any part of our bodies that's not blood or lymph in our lymphatic system is going to have, they're going to contain some microorganisms. And so our bodies are just walking around with trillions and trillions of microorganisms in addition to the cells that make up our bodies. And we're starting to understand how this influences our health and also disease, and how some of these microorganisms are actually really important for maintaining our health. And some of them, a very small minority, cause disease. So when you think about the human body, you can think about the cells that make us up, meaning the host, cells that make up the microbiome, meaning the microorganisms, and then this interaction between those 2 things and the environment. So the environment means everything we interact with, the food and water that we ingest, the air that we breathe, the things that we touch, the things that we hear, all of the environmental cues that go into our daily lives. Okay, so with that sort of context, I want to talk to you a little bit about inflammatory bowel disease. And really, this IBD is a group of disorders involving a chronic hyperactive immune response. Okay, so our immune systems are made up of different types of cells that sense and respond to invading pathogens or invading microorganisms, or some inanimate things that they know just shouldn't be there. So like if we get a splinter or something like that. And the function of the immune system is to keep our bodies protected from other organisms that would tend to grow and outcompete and make us sick. Okay, so even cancers, right? Cancerous cells aren't supposed to be growing as rapidly as they grow, and our immune response helps to keep those at bay. And so anything that would pose a threat to our well-being, our immune system has evolved to sense and respond to. Okay, so IBD then is the clinical phrase to sort of describe all these gastrointestinal disorders where there's a really hyperactive and overreactive immune response, and it's chronic, meaning it happens for a long period of time. So this comes from a website called the Crohn's and Colitis Foundation of America. And this website is it's a large organization, but primarily hosts a lot of information and research about IBD and these types of disorders. The two main types of inflammatory bowel disease is Crohn's disease and ulcerative colitis. And that's why this website is called Crohn's and Colitis. But on here, they list some examples of why IBD is such a big issue and why I'm sort of talking about it here in this lecture. Okay, so for reasons yet unknown, IBD is largely a disease of the developed world, so principally the US and Europe. So that's kind of curious, right? Because usually in developed countries, we think of diseases as being more rare, say, than underdeveloped countries that might not have access to healthcare. But IBD is actually the opposite. IBD you see more in developed countries than in underdeveloped countries. Similarly, Crohn's disease and ulcerative colitis are reported to be more common in urban than in rural areas, and in northern than in southern climates. So here's another sort of a very peculiar observation where urban settings where the hospital might be right around the corner, where there's a lot of emphasis on sanitation and cleanliness, why they might have a higher rate of disease is kind of curious. And then finally, frequency of disease increases when populations move from underdeveloped to developed countries and vice versa. And this is really important. This is sort of it's an observation, but it's very profound because this is how we would do science. We would say, okay, is this really an effect of where somebody lives, or is it an effect of the person themselves? But when people from an underdeveloped country move to a developed country, then we see the incidence of disease, or how often that disease happens, we see that increase, and vice versa. So when folks from developed countries move to underdeveloped countries, we sort of see the disease incidence go down. And that's very profound. And put together, these sort of observations suggest that there might be an issue going on in developed countries that promote IBD. So one of the hypotheses, or one of the guesses of why this might be true, is called the hygiene hypothesis. And this hypothesis states that raising children in extremely hygienic environments, sterile environments, or environments that a lot of sanitation is taking place. Raising children in this sort of extremely hygienic environment increases the risk of immune dysfunction later in life. Okay, so what that means is that the chronic hyperactive immune response in a lot of IBD people could be, in fact, a result of how those children were raised and possibly being too clean. Okay. And that's just a hypothesis. It's just a guess. It doesn't mean that it's true. It's just one way to explain these observations. So, so what's the evidence? Is there evidence out there that would support this? Well, first of all, let's look at sanitation. Okay, so sometimes this is sort of called the health revolution, but basically, what was noticed back in the 19th century and the 1800s was, there's a lot of death. The death was all around, right? A lot of people suffered from infectious diseases. There was a lot of death at childbirth with women and infants. And so there was a lot of emphasis put into trying to decrease these deaths right about the turn of the century in 1900. Okay, and right about then is when the United States, at least, developed health departments. So these were local departments of health that we still have today that look at how to make or how to decrease infectious diseases, how to decrease the mortality rate in local communities. Okay, so that was a pretty big emphasis back in the 1900s. It was noticed that a lot of infectious diseases were spread through water, and so you have chlorination of the water, and that had a big impact on health and decreased the mortality. And then you can see spikes. Here's the flu pandemic, the 1918 pandemic. Similar to what we're going through now, you can see a big spike in the number of deaths per 100,000. And if you did this 20 years from now, we can look back on SARS-CoV-2 or COVID-19, you'll definitely see another spike similar to this one. But you can see that overall this line goes down, 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 down. You have infectious disease like quarantining of folks where they can't transmit things like the bubonic plague. You have antibiotics, you have vaccines. All of these things help decrease the number of people that die per year in the United States. Okay, and so you can see that from the 1900 benchmark all the way to 2000, so across this 100-year span, we really did a lot in this country to decrease the number of people dying from preventable diseases. And a lot of that was due to sanitation practices, putting chlorine in water, using fluoride toothpaste, using antibiotics, using vaccines. All of these are sanitation practices that ultimately had a big impact on health and well-being in our country. But that said, if you look at the incidence of IBD, and again, the two major types of IBD are Crohn's disease, and Crohn's disease maps are shown over here on the left, and ulcerative colitis, and those maps are shown on the right. And these maps are such that the series A, B, and C are at different time points through the 20th century. So maps A, both map A's are pre-1960. Then you have map B, and this is 1960 to 1979, so about a 20-year window. And then map C, which is 1980 to 2008, which is about a 30-year window. But anyway, you can see that the incidence of IBD was opposite of those mortality curves that I just showed you across the 20th century. And if you compare, for example, the United States up here in panel A, down to panel B, down to panel C, in both ulcerative colitis and Crohn's disease, the incidence of IBD went up, 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 and up. Okay, until now, currently, we have a very high risk level in this country of acquiring IBD, some of the highest in the world. Okay, so those are the data that support that hygiene hypothesis, meaning that as this country became sanitized, as we did all of these things to clean up our environment and decrease infectious diseases, the incidence of IBD went up. Okay, so there's also a more specific application of the hygiene hypothesis with IBD. This is called the IBD hygiene hypothesis, proposed by Joel Weinstock. And he asked, okay, if this is true, then we should be able to go back and look for what bugs or microorganisms were present back before the sanitation practices that might be missing now. And one of the things that he looked at was a microorganism, actually a parasite called soil-transmitted helminths. And if you look at the incidence, okay, where these STHs are today in the world, and this is a little bit of, these data are a little dated, but you can see that the countries where these parasites are a public health concern, meaning they're at a pretty high high incidence, or a lot of people in these countries have soil-transmitted helminths. You can see these are the equatorial countries, a lot of underdeveloped countries in these red, the countries shown in red. And as you move into the northern latitudes, these are countries where they might still be transmitted, but they're definitely not a health problem. And then the green countries, the most northern countries, where they're not a problem at all and they're not even transmitted anymore. Okay, so this pattern kind of fit the hygiene hypothesis, where these soil transmitted helminths are in underdeveloped countries and not so much a problem or even transmitted in developed countries. So what are soil transmitted helminths? These are, again, these are parasites, so they're not good for us overall. Human soil transmitted helminths generally fall into three categories or three names, three different species, three or four, I guess. But they're roundworms, whipworms, and hookworms. And you can see that in this table that a lot of people are infected. So these are in millions. And so this is actually 1.5, about 1.5 billion people are infected. Between 1 billion and 1.5 billion people are infected with these three different types of soil transmitted helminths. Okay, so of billions of people that are infected, only hundreds of millions of people, okay, so significantly fewer, will ever know that they have it. And morbidity just means that you have some kind of symptom, and that symptom might be, lethargy or weakness, or maybe you have some cramping, gastrointestinal issues. There's some kind of symptom related to having these parasites. But of the 100 millions of people that even know they have it, only tens of thousands of people actually die from these every year. And most of these deaths are in children. These parasites definitely play a role in malnutrition and interact there. But I just want you to appreciate that of the billions of people that are infected, between 10 and 60,000, according to this table, die from these parasites. So there's a lot of people here in the middle that either have the parasites and don't die from it, but have symptoms, or that don't even know that they even have it. And so this again kind of supports the hypothesis that these things were, yes, they were parasites. Yes, they're not good to have around in such numbers, but maybe they did something to our immune systems that helped us down the road and helped us deal with things like infectious disease. Okay, so what could be the mechanism? And there's a few mechanisms listed here. These things could directly interact with different cells in our body. I just want to draw your attention to one of the ones that our lab is interested in, and that is whether or not these helminths actually interact with the bacteria that live in our gut. And that's sort of this, what they call the flora. This was the term given the gut microbiome back in probably the 60s or 70s, but today we call this luminal flora, we call that the microbiome. So one of the questions that we asked in our lab was whether or not this helminth could influence microorganisms in the gut and change the way that our bodies respond to infectious disease. So what we did in a few different experiments that we published was look at the bacteria in our gut that live or get that increase in abundance actually after these soil transmitted helminths infect a host. And in this situation, we're using mice. We're using a mouse model and a soil transmitted helminth that infects mice. And we infected a group of mice and we asked, what bacteria increased in abundance during this infection? And one of those that we found was called lactobacillus. And lactobacillus might sound familiar to some of you, especially if you're into probiotics. There's a lot of lactobacillus types of bacteria or related bacteria in probiotics. And so we thought, well, that's kind of interesting. If the microbes that get that increase in abundance that do very well during infection are ones that also are purported to have some anti-inflammatory effects on humans, that could be very interesting. And we published this, and it was actually looked at again by another study a few years later, and they did the same thing. They infected some mice and compared those to what the microbiome looked like before they were infected. This was in the small intestine, And in the exact same spot where we looked, indeed, they found that this big chunk of the pie over here at day 14 was comprised of lactobacillus-like bacteria, and that they really increased from baseline. And that was really, nice because it supported our data, but it didn't really tell us what those influences might be to the host. There was a third study published in 2014 that found the same thing. I'm not going to dwell on this, but basically these mice have a higher level of lactobacillus than the uninfected mice. So 3 studies now have supported this. And it was this third study that said, okay, as the number of lactobacillus increase, so as these numbers go to the right on the x-axis, what happens to a certain immune cell that we know is important in IBD? And this immune cell is a regulatory immune cell. It helps to regulate our immune response, meaning calm it down if it gets too active. And you can see that as the number of lactobacillus go up, so do the number of these regulatory immune cells, suggesting that this is indeed a really nice trend where you can have a parasitic helminth infect your GI tract and encourage bacteria in your gut that then feed back and tell your immune system, hey, you need to calm down. And that's really an interesting part of this dynamic that our lab is continuing to look into. So this didn't end there. There's a lot of folks that are interested in coming up with new treatments for IBD. These are chronic diseases that folks have pretty much for their whole life after they get diagnosed. There's no cure currently for IBD. So symptoms, we really need better treatments for these symptoms. And some of those are to use and manipulate the microbiome, the bacteria that live in the gut, either by giving them helminths that don't make them sick or would kill them, or by giving them certain members of the microbiome like lactobacillus that might make them better. And right now in the US, there's ongoing clinical trials for IBD. I just looked today, and there's actually, I think 6 or 7 clinical trials that are ongoing for IBD that use helminths or something like a parasite that would that they're testing whether or not it has a benefit in people. But other types of hyperactive immune response or disease that's linked to hyperactive immunity, like autism, psoriasis, multiple sclerosis, even allergies. There's ongoing clinical trials that use helminths or something from a parasite to treat people. And so this is a very active area of research that has direct links to how we've changed our environment, how we've sanitized it. Okay, so those are the examples. I just want to talk a little bit about some of the biotech that we use in the lab. So a lot of the work that we do requires some kind of control, right? So when you're looking at a mammal like a mouse, all mice have a gut microbiome. So what do you compare those mice to? Say if you wanted to do a manipulation, or if you wanted to give them, say, treat with an antibiotic, or you wanted to test for the function of just one individual of that microbiome, you would be really hard pressed to come up with a study design that would help you look at that. So one of the inventions over the past 100 years has been germ-free animals. And these are animals that are born completely sterile, and they live their whole life without any bacteria or other microorganisms. And so you might ask, how can this be done? Well, we're all born sterile, or we're all we all develop sterilely, at least in the gut. We all develop sterilely in our mother's wombs. We don't have any bacteria in our gut. We don't have any bacteria in our lungs. We don't have any bacteria anywhere. And we stay that way until gestation, or until we're born. And we can be born naturally, and we pick up those bacteria in the vaginal tract of the mother, or we can be born with C-section and then pick up bacteria and microorganisms in the environment right after we're born. Either way, we develop sterilely. So germ-free mammals, at least, can be born at gestation using a sterile surgery like a C-section, and after birth, they can be suckled in these isolators, and they can remain that way. They can remain sterile. So let's just talk through this. You can actually buy germ-free animals. This is a shipper, just to show you what some of these look like. We had some mice that we ordered in from another lab, a vendor. And you can see that here's a mouse in a cage. And this cage has a lid with a HEPA filter, so a filter that prevents any microorganisms from getting in there. And then this whole cage is inside a hermetically sealed isolator, and on the end over here on the left, you can see this white filter. That's again a HEPA filter that allows air to come in, but only at a particle size that limits microorganisms. So they come sterile right from a vendor. But we can use these chambers shown here on the side and on the back to raise these animals completely sterile their whole lives. So here again is our shipper that I just showed you on the cart here, and we can port that up to this chamber through a port door. And you can see this port kind of looks a little gray, kind of looks a little cloudy. And that's because after we port those up and before we move the animals through, we have to aerosolize that port door. And we do that with a chemical sterilant that completely sterilizes the inside of that chamber. After we get that thing sterilized, then we can bring the mice through in the cages. We bring them into the isolators, and we can give them fresh food and water. We can change the bedding. We can do all of that inside the isolators. So that's how we sort of take care of these mice. But we can also derive them germ-free doing the sterile C-sections from mom. We do that routinely as well with biosafety cabinets and other kinds of engineering controls in the lab. Here's one that we set up. So you might wonder, how do we make sure that there's absolutely no microbes in there? Well, we have a couple of different ways to do that. Right underneath all of our cages, we have what we call media traps. So this is just water that contains a little bit of the food that the mice eat. And if you put that food in the water, it turns sort of this color, this orangish color. And that orange color just lets us know that there's enough nutrients in there for microbes to grow if there are any. And we monitor that to see if it becomes cloudy. It would be the same as if you took chicken soup out of a can, you put it into a pot, and you watch that pot day in and day out. There would be microbes that grow in there very quickly. Probably the next morning you'd wake up and see, hey, this doesn't look so clear anymore. So that's exactly what we're doing here. We're just monitoring for growth in these things. And then we can take samples out of these, and we do that about every other week. So every 2 weeks, we'll sample our traps in all of these isolators, and we'll look for microbes with those DNA tools that I mentioned at the first part of this lecture. And we'll ask whether there's any microbial DNA there at all. And if there is, then we know, okay, we had a breach in sterility, and that triggers a lot of work for us. We have to decon the whole thing, take all the mice out, move those to another isolator. Sometimes we can still do experiments with those animals, but we have to decontaminate all of the isolator before we can get it up and running again. So these mice really give us a clean slate, and we can add back into those mice exactly the microbes that we're interested in. Say we want to look at E. coli or lactobacillus or something else in the gut microbiome. We can give those mice just those microbes that we want to look at, and that really lets us do these types of experiments. Okay, so I'm just going to end today with a few interesting questions. If you haven't thought about these, they're kind of interesting about your own bodies. Okay, so how big is your GI tract? This is probably going to be the question for your D2L quiz or attendance today. So I'll give you some some options there. Some folks say it's like, maybe the size of a house, a badminton court. If you don't know what a badminton court is, I guess you have to Google that. So just remember for this one, I just want to give you a little clue that inside our gut, we have villi. These are invaginations that go up. They're finger-like projections into our gut lumen. And if you drill into these villi down to the single cells, you'll see the single cells have these microvillus structures that are on top of them. So we have these small villi on top of large villi. So just think in your head about what that might do to the size of our GI tract as far as surface area goes. Okay, I'm going to skip this one, but I'm going to talk just briefly, let you guys know that we are doing some really cool things in the lab. Like we're developing tissue cultures that closely resemble that gut structure. So the luminal structure and microvilli of the gut that I just showed you, we can grow tissue in the lab that resembles these structures. So this would be the gut lumen. Here we're looking at the surface of one of these organoids that we're growing in the lab. And these are what they look like actually under the dissecting scope. They have these little blebs on them. And these are like little guts in the dish that we can grow. And we can infect these things. We can culture bacteria inside, and those bacteria can grow right on the surface of the gut, just like they would in our bodies. And we can also put in immune cells into the dish. So around the outside of this gut, you can see these purple or blue cells. Those are actually dendritic cells, or I'm sorry, they're macrophages that we put into the culture around this little gut in a dish. And then inside in green are microorganisms that we're growing there. So we have our immune system, our gut, and the microbes all growing together in a dish. And that's really another biotechnologic leap that helps us look at the gut microbiome. Okay, so that's all I have today. I'll be happy to take some questions. You guys have.
Well, if you have a germ-free mouse, does its poop smell?
Yeah, that's a great question, Dave. It actually doesn't. And that's another way we can monitor for any kind of contamination. It won't smell. And actually another thing is every once in a while, if you're raising these animals inside a chamber, one of them will just die and you don't know why, but sometimes it happens. And anyway, if that mouse is completely germ-free, it won't bloat up. You know, like we see these animals on the side of the road, somebody hit with a car, they're all bloated. Yeah, these animals, the bloating in normal, when you die normally, is due to microbes and the gases that microbes make. And so these mice in these chambers, if they happen to die, they don't bloat and they just kind of shrivel up as the water, as they dehydrate, they just kind of shrivel up into nothing. It's really weird.
Juan? Sorry. Go ahead and talk.
Have any of these mice developed IBD when there aren't any germs?
That's a great question. Yeah, so have the mice developed IBD without any bacteria inside? The answer to that is no, basically. So there are a few different models of IBD that we can use in the lab with mice, and a lot of these models are dependent on bacteria being in the gut. So when you have a germ-free animal, they don't get disease, they don't show signs of hyperactive immunity, and other signs that we use to tell us, yeah, there's a problem. Yeah, it's completely dependent on microbes being there.
So we have another one here.
What causes a mouse to just drop dead in a sterile environment?
What causes the mouse to what?
In a sterile environment.
Yeah, I'm sorry, I didn't get the question. What causes the mouse to what in a sterile environment?
Drop dead. What causes
Oh,
a mouse to drop dead in a sterile environment?
yeah. So these mice, these lab mice that we use, are inbred. They are they're bred that way, so there's not a lot of genetic diversity that influences the diseases that we look at. So every once in a while, there's just a something about the mouse that is not just not working right, and it's not. It doesn't happen very often, but it just randomly happens that you're going to have some mice that die, and you just don't know why.
I have a more general question. It has to do with antibiotics. Most of the antibiotics that I know about are taken from microbial cells. Is that true?
That's very true. Yep.
And these are microbes that grow, but we hear that 95% of the bacteria in the soil do not grow. But do you think they produce antibiotics?
Uh, so most microbes have defense mechanisms against other microbes. So a lot of bacteria make phages or bacteriophages, and they're little viruses that go and kill other microbes. Some of those are very closely related, others are not. And so viruses you could think of as antibiotics as well. The microbes that you're talking about, Dave, that make antibiotics, they're making more complex compounds that would then go and kill other things. So yeah, I think microbe interactions are fascinating, and oftentimes microbial communities limit themselves rather than the environment limiting the microbes. If that makes sense.
Could we mine the DNA of the organisms that cannot grow in culture in hopes of finding a way to find new antibiotics? Somehow clone the DNA into a cosmid, something like that? You have any
Yeah,
idea about that?
yeah, that's being done. It's been done in the pharmaceutical industry for quite some time. There are a lot of drug screens out there, not just for antibiotics, but for cancer drugs and other types of diseases. You might want a drug that influences a human cell, so you grow that human cell in a dish and then see whether it can be effective. But yeah, for microbes that you can't grow, you can still work with that DNA sometimes by capturing it into plasmids, and then you put that plasmid into something like E. coli and have the E. coli make it. Yeah, so those things are definitely being done.
We have time for one more.
Are there any other animals that have gone through the same sort of decontamination as these mice? And if there are, how did that affect the way that these tests go?
Yeah, great question. The first germ-free animal was actually a quail. So birds are really easy to make germ-free. You can take their eggs and you can dunk them in a bleach solution and then culture those under a light. And birds, you don't need to breastfeed. You know, they sort of eat food right out of the gate. So those are very easy animals and have been used. But there's also germ-free calves, so cattle, germ-free pigs. They made germ-free marmosets as a small mammalian model. Yeah, so there's a lot of different germ-free and gnotobiotic animals. Now, when you're raised completely sterile without gut bacteria, there are issues with nutrition, right? Bacteria really help us get what we need out of our diet. They help digestion. So a lot of these animals have trouble digesting diets that other animals with the microbiome would eat normally. So we give them special diet. We try to overcome some of those problems with by changing their diet.
That's almost the end of the lecture, but I'm putting together what I will call a added value exam. It's going to be given maybe on next Monday, and you'll have a week to answer the questions. It's just an exam to see what you guys are thinking now that we're 1/3 of the way through the course. Thank you very much, Seth. A beautiful
You got
job.
it. Thank you, guys.